This video is going to cover section 12.2, which is on the transmission of nerve impulses. So the nervous system, it uses the nerve impulse to convey information. And the nerve impulse can be studied using excised axons and a voltmeter called an oscilloscope. So the voltage is measured in millivolts, and it's a measure of the electrical potential difference between 2 points. In a neuron, the 2 points are inside, in other words, in the axoplasm, and in the outside of the axon. On a voltmeter, the voltage is displayed as a trace pattern over time. The resting potential in an axon that is not conducting an impulse, the voltmeter, it would record a potential difference across an axon membrane equal to about minus 70 millivolts. So in other words, the inside of the axon is negatively charged with respect to the outside. So this reading is known as the resting potential, and it shows that the inside of the axon is negative compared to the outside. So there is a polarity across the axonal membrane, and the resting potential is the potential difference across the membrane in a resting neuron. So the polarity is being shown here. So you have a greater number of negatively charged molecules inside the axon than you do outside. And so we have an excess of positively charged ions in the extracellular fluid of the axon. So this unequal distribution, it's maintained by carrier proteins, referred to as the sodium-potassium pumps, which we've talked about before. And these actively transport sodium out of the axon and potassium into the axon. Now, the pumps are always working because the membrane is permeable to sodium and potassium. So the membrane is more permeable to potassium, and therefore there's always more positive ions outside the membrane than inside. So negatively charged organic ions on the inside of the axon also contribute to the polarity across a resting axonal membrane. So you have excess positively charged ions outside the membrane, and then you have the negatively charged organic ions on the inside of the axon. An action potential is it's a rapid change in the polarity across an axonal membrane as the nerve impulse occurs. And this is an all or none phenomenon. So if a stimulus causes the membrane to depolarize to a certain level, referred to as the threshold, then an action potential occurs. And the strength of an action potential does not change, but an intense stimulus can cause an axon to fire more often. So it requires two gated channel proteins in the membrane. One channel protein allows sodium to pass into the axon, and another channel protein allows potassium to pass out of the axon. So here you can see the polarity of the axon, the inside with respect to the outside. So we originally have the resting potential which is about minus 70 millivolts, and then once it reaches the threshold, then an action potential occurs where you initially have depolarization where sodium flows in, and then you have repolarization where potassium flows out. It results in an overshooting, a hyperpolarization, and then back to the resting potential. So we'll look at each of these different parts in a little bit more detail here. So the sequence of events then is initially the sodium gates open, and this results in depolarization. So when an action potential begins, the sodium channel gates, they open, and sodium flows down its concentration gradient into the axon. So you have higher concentration of sodium outside than you do inside because of the sodium pump. And so therefore, when the gates open, the sodium channel gates open, then then sodium can flow into the axon. So as sodium moves inside the axon, the membrane potential changes from minus 70 millivolts to positive 35 millivolts. And this is called depolarization because the charge inside the axon, it changes from negative to positive. And so this, you can see it in this um, illustration here where the sodium channels, they open up. It allows the sodium to pass into the axon. So then after that, after you have depolarization, then repolarization occurs when the potassium channel gates open, and then potassium now flows down its concentration gradient, which is outside of the axon. So you have a greater concentration of potassium inside the axon than you do outside. And so when the potassium channel gates open, then potassium will naturally flow outside of the axon. So as potassium flows out of the axon, then the action potential becomes more negative again, which is then repolarization. During this time, it briefly becomes slightly more negative than its original resting potential, which is referred to as hyperpolarization, but that only occurs for a very short instant. So here you can see in the illustration again, the potassium channel gates open, which allows the potassium to exit the axon, and again, it's just simply along its concentration gradient. Um, Now, there is a difference between myelinated and non-myelinated axons. If you recall, in the first video we talked about the myelin sheath, and that was produced with the Schwann cells, and so we'll look at the difference between them right now. So action potentials in non-myelinated axons, um, the action potential travels down an axon just in one small section at a time. So when an action potential has moved on the previous section, it has to undergo a refractory period during which the sodium gates are unable to open, and the action potential cannot move backwards, so it always moves down an axon. And you can generally think of that as being from the cell body down the axon, um, away from the cell body itself. When the refractory period is over, then the sodium potassium pump has restored the ion distribution by pumping sodium out of the axon and potassium back into the axon. In myelinated axons, the gated ion channels that produce an action potential are concentrated at the nodes of Ranvier. So that means then that ion exchange only occurs at these nodes. Therefore, the action potential travels faster than in non-myelinated axons. So with myelinated axons, the action potentials occur only at the nodes of Ranvier. So therefore, the action potential just basically jumps from node to node, which is referred to as saltatory conduction. And so therefore, the myelin sheath allows the action potential to travel much faster along a myelinated axon, because the action potential doesn't have to travel along the entire length of the axon. The um, the ion exchange occurs only at the nodes of Ranvier. So this results in a much faster transmission of impulses along the axon. So the transmission across a synapse then, um, every axon, it branches into endings that have a small swelling, which is referred to as the axon terminal. And each terminal lies close to the dendrite or the cell body of another neuron or a muscle cell. And this region of close proximity is what's referred to as a synapse or a chemical synapse, mostly just simply a synapse. And so what we have is the membrane of the first neuron, that's referred to as the presynaptic membrane, and then you have the membrane of the nerve cell that's receiving the message, and that would be the second neuron or the postsynaptic membrane. So two neurons at a synapse, they actually do not physically touch each other, but they're separated by a tiny gap which is referred to as the synaptic cleft. So an action potential, it cannot cross a synapse. So therefore, the communication between the neurons at a chemical synapse, it has to be carried out by neurotransmitters. And these are chemicals that are stored in the synaptic vesicles in axon terminals. And you can see an illustration of it here. And we'll just explain that process, how that takes place. When an action potential arrives at an axon terminal, gated channels for calcium, they open and calcium enters the terminal. Calcium interacts with contractile proteins, which contract and pull the synaptic vesicles to the presynaptic membrane. And the rise in calcium then, it stimulates synaptic vesicles to merge with the presynaptic membrane, which results in exocytosis. And this is similar to exocytosis that we've seen in other types of cells. Neurotransmitter molecules, they're released into the synaptic cleft and they diffuse across the synapse to the postsynaptic membrane where they bind with specific receptors. And depending on the neurotransmitter, the postsynaptic neuron will either be excited, which causes an action potential, or it will be inhibited, which actually stops an action potential from occurring. So a neuron can receive many excitatory and inhibitory signals. Since its dendrites and cell body, it will have synapses with many other neurons. It's not just a simple, one-to-one connection between neurons. You can have hundreds or maybe even thousands of connections that occur between nerve cells. So excitatory signals, they cause a depolarizing effect, whereas the inhibitory signals, they cause a hyperpolarizing effect. And so therefore, synaptic integration is the summing up of the excitatory and inhibitory signals in a postsynaptic neuron. So if the combined signals cause the membrane potential to rise above the threshold, then an action potential will occur. So in other words, it's a summation, if you will, of all the different inputs that a cell is getting at one time. And if it results in a rise above the threshold, then the nerve cell will fire. Once a neurotransmitter has been released into a synaptic cleft and initiated a response, then it's removed from the cleft. And this prevents continuous stimulation or inhibition of postsynaptic membranes. In some synapses, the postsynaptic membrane, it contains enzymes that actually break down the neurotransmitter. In other synapses, the presynaptic membrane, it reabsorbs the neurotransmitter for repackaging then in the synaptic vesicles, which it can then release at a later time. Many drugs that affect the nervous system, they act by interfering actually or enhancing the action of neurotransmitters. So drugs can enhance or block the release of a neurotransmitter. They can mimic the neurotransmitter. They can block the receptor for the neurotransmitter or interfere with the removal of the neurotransmitter. An example of this is sarin gas, which is a chemical weapon that inhibits acetylcholinesterase, which is an enzyme that's responsible for the breakdown of acetylcholine. And so then this leads to a prolonged acetylcholine activity and results in convulsive spasms in the individual that the drug has been administered to. So that covers the transmission of nerve impulses along the axons, and we will continue looking at the nervous system in some more of the videos.